Giske is a professor of clinical microbiology at the Karolinska Institute at Stockholm in Sweden. His research is mainly involved on antimicrobial resistance and genomics. And he's currently the chairman of the European Committee of Antibiotic Susceptibility Testing that is hosted by the European Society of Clinical Microbiology and Infectious Disease. So we are glad to invite you and to hear your talk. The title is Extensively Drug-Resistant Enterobacteriaceae, the Role of Epidemic Clones. Thank you, Christian.
Thank you very much, Jean-Marc, for that very nice introduction. Is it okay to start now, or should we wait a minute more?
One more minute.
One more minute.
Yeah.
Okay. Let's do it.
That's it.
It's okay. It's okay.
It's okay.
It's okay. Okay. Wonderful. So thanks a lot for inviting me. It's a pleasure to be here. It's a very impressive facility, obviously, and it's really nice to see the place and Marseille as well, which I hope to see a little bit more of later today. Just to briefly introduce myself, I work 40% in the hospital, and then I work 60% in the university. And then I have the extracurricular activities on the other, 30–40%. So basically, a lot of my work is now spent with the European Committee on Antimicrobial Susceptibility Testing, probably at least 10 hours a week, and quite a lot of traveling to also sometimes remote places. So that's a quite tough challenge sometimes, but also very much fun in many ways. I work also as a senior advisor microbiologist in Stockholm County, so I try to work with different groups dealing with the antimicrobial resistance and infection control and other issues for Stockholm County. And then finally, I also spend some time doing antimicrobial surveillance, basically together with ECDC and also with Public Health Sweden, where one of my PhD students actually also works. And I'm also monitoring what many of you may be aware that the CLSI is the American counterpart to the European UCAST. So I'm monitoring what they are doing as well, trying to visit their meetings once or twice per year in the US, and getting into the different aspects of antimicrobial susceptibility testing. This is where we are located. We have a new hospital that has been recently opened. It has a lot of technical or IT problems, basically. But they are trying to solve that. It's anyway a hospital consisting mainly of single rooms, like you have in the ID department here, but not all of them with external possibility of entry. The university part is located at the south campus of Karolinska. It's both the hospital and the university are located in 2 geographical sites. And so this is where most of the research activity is ongoing, but I also have some labs within the hospital. So it's a little bit of a mix, and I actually moved geographically when I moved from university to hospital. These are some of the people in my group. There are also other groups at the same place here, some of them working with antimicrobial peptides, some with virology, different things. Senior Professor Carl-Erik Nord, who is an old friend of Professor Raoul here, is one of the group members, and also a couple of new postdocs. We have a research funding from a variety of sources. So we have one EU project running now, and we have also some other funded from Eurostars, and one from this joint programming initiative in antimicrobial resistance. So right now at the moment there is some momentum to expand the activities and try to look into some of the problems that we will be discussing here. I thought I would start by actually a little bit more philosophically enter the topic by asking, when does antimicrobial resistance really matter? And mostly that depends, of course, what you mean by when does it matter? But if you mean for mortality, it's quite natural that it will only have a significant meaning in case of severe disease. And even in severe disease, it's quite frequently difficult to assess what is actually the contribution of AMR to the mortality. There are so many confounding factors, and one of them being the patient comorbidities. Many of those infected by very resistant strains, they can also be infected by fungal pathogens. They are severely immunosuppressed, they have other underlying conditions. So whatever they die from is not necessarily from antimicrobial resistance. There are also some types of resistance that come with greatly reduced bacterial fitness, and I would say that the role of strain factors is usually very insufficiently studied. So it's not only about the AMR, it's also about the strain that infects you, and of course the host. So if you look at other diseases, it will not be meaningful to use really mortality as a parameter. You would need to look at length of stay or increased costs for the hospital. You could look at impact on strain transmission and other long-term consequences. But we tend to look mostly on bloodstream infections because they have more of an undisputed relevance. It's very easy to obtain the strains. They are always saved, so you can work with them later on. And it's also easier to compare between the countries because other infection types vary even more than bloodstream infection in the clinical management and in the microbiological management. So there are also some things to be said about in vitro resistance versus clinical response. And it's quite evident that clinical response and in vitro susceptibility testing is not a very good match. I mean, the testing is the best we have, but it's not very good. And we know that if a strain is susceptible, it responds in around 90% of the cases. If it's resistant, it still many times responds in almost 60% of the cases. So these are quite crude tools we have, but we still use them, and in terms of understanding the burden of resistance when we are not necessarily talking about the patient, I think we can still use the methods, but knowing that we are, of course, simplifying the true situation. And also these numbers I mentioned here mostly pertain to monomicrobial infections when there is a predictable penetration of the drug. So if it's polymicrobial and unpredictable penetration, it could be even lower than this. There are also all of the factors related to strain variation, as I mentioned, and this, for instance, that strain virulence is not taken into account in AST is quite important. If you look at preclinical PKPD, the killing of bacteria, it varies extremely much what is actually necessary of antimicrobial exposure to kill a strain, depending on what strain you look at. So it really is, you could say, all the things I'm doing and spending 10 hours a week on is actually quite weak evidence behind. It's not great science, and still I put time into doing it because I think we need to work with the tools we have. But there are so many things to be said about this, and when we now return to talking about the resistant strains, we should know that there are plenty of things to be said about what this actually means for the patient. When we look at bloodstream infections, it looks very similar in different European countries, and most of the time we find that E. coli is always the most common. It does to some extent vary, but usually Staph aureus is number 2, and then it varies which one is number 3. It could be Enterococcus in some places, and it could be Klebsiella in other places. And there are even countries that have a very skewed species distribution in blood cultures, such as, for instance, Greece, which has a lot of Acinetobacter, but probably this is down to sampling practices and that they are not always sampling community-acquired infections. But I will focus this mainly on Klebsiella and E. coli because they are the two most common among the gram-negatives, and this is also where we are seeing a lot of the resistance problems. So if we start then by looking at Escherichia coli, This is maybe not the map you're used to seeing, because this is a map also including the areas that are covered by another surveillance network, which is the CSER network, the Central Asian and Eastern European Surveillance of Antimicrobial Resistance, and that's actually those countries plus Switzerland. And that sounds a bit peculiar, but this is because Switzerland does not want to pay anything to ECDC, and thus ECDC does not want to receive data from Switzerland and put it on their maps. But fortunately, we have this a possibility of looking at all of the data when we actually then turn to merging the data from the Central Asian and Eastern European system with the one that's hosted at ECDC. And we can see that there is an upwards trend for ESBL-producing E. coli, and this is also one of the areas where even in the Scandinavian countries, the resistance level is slightly higher. It's not below 1%. There is a gradient, and many of the countries further to the east and to the south tend to have more resistance. We know that this is due to the stepwise development of different beta-lactamases, starting from the pan-susceptible through the penicillinases, through the first types of cephalosporinases, and then the CTXN variants, and finally the carbapenemase. So this is a development that has been observed over a long time, and moreover, we are seeing also that lots and lots of individuals in some areas of the world, and even in Europe, quite a lot of people carry these strains in their gut microbiota. In some of the Southeast Asian countries, it's more common to have ESBL carriage. It's at around 70% almost in some places. So it's more common to have carriage than to not have carriage. In Sweden, we did an investigation of 2,000 individuals from the general population. This was published by Peter in my group, and we found that there was around 5% carriage. So given that there is 5% of the population actually carrying ESBLs, it's evident that some of them are going to have bloodstream infection, but it's not evident who they are going to be. With regards to carbapenem resistance in E. coli, it looks better. There are some countries, for instance like Turkey and Greece, which have moved into 1 to 5% instead of less than 1% resistance. And if you look at this year's, well, this is not this year's, but there is always 1 year delay. So this is the most recent published now in 2017, showing that even more areas have moved into 1 to 5% resistance in E. coli. But large parts of Europe have very low resistance to carbapenems in E. coli. Turning then to Klebsiella, this is a much more concerning situation in most cases, in most countries. But if we look at the Scandinavian countries here, they actually have, even lower occurrence of ESBL-producing Klebsiella than of ESBL-producing E. coli. Probably this is due to the fact that this is a hospital pathogen. It disseminates in the hospital environment, and one of the reasons is then that if you have a low transmission in the hospitals, you also don't see very high rates of this type of resistance. It's a very clonal type of resistance. You have a few clones that will spread quite substantially, and then suddenly your country will turn into an unpleasant color, in this case with very high levels of resistance. And this goes also for carbapenem resistance, where we see that we have this problem that has been known for a while in Italy and in Greece, but we also have Turkey, and we have Russia starting to come in here as well. So there are plenty of countries who have problems even of the carbapenemase-producing Klebsiellas. And what's also concerning is that 1/3 out of these are also resistant to polymyxins. So that means they are double resistant. You could call them CCRE if you like, Colistin and Carbapenem-resistant Enterobacteriaceae. So that's quite concerning from a treatment point of view. This is a situation in 2016, which looks, the colors are moving to the higher resistance levels. And happening here, and this is something that's relating to the rest of the talk. And this is just to remind us about from this survey that Harry Grundman did on behalf of the CDC, which was published this year in Lancet Infectious Diseases. I was also involved in this work, and you can see that the KPC was the dominating genotype. OXA48 was the second place, and also one can see a bit which genotypes were found where, and basically most of the genotypes were found in most places, with some exceptions such as DIN, which is found mostly in Spain and in Greece, and not so much in other places, with the exception of Hungary maybe also. Even in this collection, it was found that 40% of these strains were also colistin-resistant. So that's quite concerning, and if we look at the new drugs we have and what they are actually susceptible against, we have had recently cefotaxime-resistant, which really is only active against ESBL-producing Enterobacteriaceae and also multilocus Pseudomonas. For the others, it doesn't have any useful activity. Cefotaxime-resistant covers also to some extent KPC, but it has limited activity in Pseudomonas. Now we have Meropenem-resistant, which is the first carbapenem combined with the beta-lactamase inhibitor, and there is also in the pipeline Imipenem-resistant and some other drugs which will not be mentioned, but actually the only one that has activity against all of these problem pathogens is still colistin. And that's why colistin, even if it's not a very impressive drug, it still has a significance, and moreover, there is already resistance to some of these new agents. This is a paper that was published this year in a patient who had received meropenem therapy, and where they could select the strain that was resistant to cefotaxime-resistant. And that was due to foreign mutations and increased expression of KPC3, which was resolved by PACB biosequencing of the strain before and after. So this is clearly something that's, we are going to be relying on colistin for some more time. We have the problem of the mobile colistin resistance, which I know you have been working on here as well. And this is of course something which is largely coupled to animals. So here it's quite clear that this is a very important source. Most of them have been described for the first time in animal isolates. And we know that they use extremely high amounts of colistin within animal husbandry. So this is clearly one of the main factors, although it would have to be said that most of the strains I showed you, which are carbapenem resistant, are not really resistant by this mechanism, but rather by the mechanism of having chromosomal mutations. But now there will be next year a big European study in which Public Health Sweden will actually also organize this, where it will be looked into specifically the carbapenemase-producing strains isolated in different countries, and they will be subjected to colistin mechanistic studies to understand not only the proportion but also the mechanisms of resistance, and even how this is the clonal structure of the colistin-resistant strains. It's quite clear already now that if you have a colistin-susceptible microorganism, you have a better chance of survival than if you have a colistin-resistant strain. So regardless of whether people think that this is a poor drug, it still seems to be doing something useful in terms of saving lives. So let's move then to the bacterial clones. This is something that may have had quite a number of definitions in science fiction and in movies and other places. I would say that probably this is again debatable how to define it, especially in the area of genomics. You can debate how to define clones and subclones, but we will look a little bit into that. But let's start by addressing some of the historical aspects of this. And historically, the first method used for this purpose was really Multilocus enzyme electrophoresis, which was not a poor method, but it was difficult to standardize. it had all of the intra-laboratory variation problems, and therefore, instead, multilocus sequence typing was introduced. And all of those who have done that knows it's quite laborious because there's a lot of PCRs and sequencing. But of course, now you can do this in silico from genomic data, which has simplified it a lot. It became quite clear very early on that it was difficult to visualize the population structure with traditional trees, and this is why they needed to look into other options. I assume that most of you are probably familiar with the MLST, but just in case some are not, you start with the genomic DNA, you have the amplification of 7 housekeeping genes, and then you give them these arbitrary numbers depending on which allelic variant they have, and this allelic profile comprises a sequence type. And these are also arbitrary numbers. You cannot really tell from the number, it's just when it's described the first time in the database. And this can be done now in silico. Traditionally the definition has been 6 shared alleles. And that means if you have this very simplified example, isolate 4 would be outside this one. You would have in the middle this strain, and on both sides of it you would have 2 single locus variants. And together they would be in one clonal complex. But this is what happens if you have few data. Obviously, if you have much more data, it becomes a bit more cumbersome to show how they are related. One of the important aspects of this emerging from the introduction of MLST was that before that we have been talking about, so in Spain we have problem with PFG clone A, and in Belgium there is problem with PFG clone H. And it was difficult to know that PFG clone A in Spain and PFG clone H in Belgium was actually the same sequence type before you had that tool. And when that was introduced, and before the genomics, I should add, one started to introduce the concept of high-risk clones, which should be associated with resistance and also high ability to spread or to confer invasive disease or to colonize for a long time. It's a question what to call them. I prefer myself to call them epidemic clones, because the only thing we have shown really is that they occur more often. We know that they occur more frequently than others, but we do not usually know whether they actually have more of a risk. The only risk we know is that it will disseminate more. But quite frequently we don't know so much more, and this I will come back to what we actually do know. I was involved in some of the initial work myself on using the MLST technology to start to characterize resistant strains. This was some work we did in 2006, where we looked at the sequence type that was just introduced then. There was an MLST protocol that just became available. And we called them back then burst groups, but later on these groups were identified as clonal complex 235 and 111, which are still among the epidemic clones that are very common in Pseudomonas. This was a work that was done together with the Gianni Rossolini's lab, so it included quite a number of Italian isolates as well. Then we did some other work in 2009 together with CDC, where they had this outbreak of Klebsiella pneumoniae carbapenemase, and they had PFG results, and they had found that they had one major clone which consisted of 172 isolates, and most of the others were unrelated. And then when we started doing the MLST, we identified sequence type 258, and that one became a very famous, or rather maybe notorious sequence type, because that's still the one that's most common, either that one or the single locus variants of this one. So with this in mind, so how can we now use this to understand something more about the population structure in bacteria? Well, partly, one thing to recognize is to know the regular population structure of the species you're talking about. You need to know if you're talking about, for instance, Staph aureus, or even to some extent Acinetobacter, which tend to have more of a clonal structure, which will over time diversify by single nucleotide polymorphisms, and you can detect those SNPs, and there is not a lot of recombination ongoing, and that means it's quite easy to follow the phylogeny. Then you have the freely mating populations where you have all sorts of recombination ongoing, and this is quite common, for instance, in Enterobacteriaceae. And what's typical is that you see this pattern of isolates which are not forming a traditional branch system, but more of this network indicating that they are recombining. You can, of course, when you do the analysis, you can subtract the recombination component and make it more easy to see the patterns. But what's more interesting really is to show this sprouting phenomenon, and that's what we mean when it's said that some of them have an epidemic population structure. It happens so that at some point there is a very successful variant emerging, and this variant gives rise to a lot of offspring single locus variants that are still related to this one. And for some reason, which we do not really know, this becomes then something that can spread in a more epidemic or even pandemic fashion. This is what we can see if we look at all of the data. This is actually one of the papers that introduced the term high-risk clone. And this is where Neil Woodford's group looked at some of these clones that I've already mentioned, 258 for instance, and some others, sequence type 11 and 14, which will be mentioned later on as well. And these were occurring more often, while all of the single dots here means it's just a sequence type that mainly occurs just on one occasion, and it doesn't occur in any other variants. So that would be an example of all of those that do not have this sprouting phenomenon that you could see on the previous slides. This is the type of diagram, the burst diagram based upon related sequence types, and of course this does not tell you the whole truth. It just tells you based on the 7 genes and the simplified types. So it doesn't really even go into how different they are if they differ in one allele. If you want to look at that, you can look at the concatenate sequence. But these diagrams are not really for that purpose. They are more to visualize this in a snapshot of the population. So that's more or less what the purpose of it is. Some have also introduced this possibility of using Google Maps, where you have these burst functions built into this. This is something that the Sanger Institute has worked quite extensively with, developed quite extensively for MRSA, for instance, where you can go over the map and you can look at what particular clones they have found in some areas. But obviously that takes quite a lot of work to do this type of thing. One popular way of showing things is to produce it in minimal spanning trees, and in these trees you will typically have around the clonal complex, you will have a gray zone, and you will have an area of each circle corresponding to the number of isolates that will have that type. That can be also a way of showing whether you have some relation, if you want to show graphically the relation between different serotypes, virulence genes, phenotypes, or even mortality contribution. You can paint them, and you can see if you see patterns that become evident for the eye. Also, you can do diagrams which are based on the concatenate sequences, and usually they will cluster them in a similar manner to this other one. So let's look then at some of the classical examples of successful gram-negative clones. We have mentioned 258 already, which I will look a little bit more closely into. But one of the other ones, which is really important for the spread of ESBL in E. coli, is sequence type 131, which is a uropathogenic clone that acquired CTXM And in quite a lot of countries, this corresponds to even around 30 to 40% of the cases of ESBL-producing E. coli. It occurs pandemically. Quite often the infections are also, although at least we do not always know if the carriage is community acquired, but we know that there is a community onset of the infection. And it has been found in meat and in companion animals. And recently there has been focus on even looking more closely into this clone, not only by MLST but by core genome MLST and SNP analysis, and showing that there is a subclone which is called H30RX, which is the most important in this context. So that's one of the clones we are quite interested in my lab. And the other one, still interested, since we have been part of the story with sequence type 8, the 258 from the beginning. And this clone really was not only associated for the initial outbreak in the US, but also for the majority of the cases in Israel. Before Israel were actually able to curb quite a lot of the outbreak. They had extremely strict infection control. They even had subtraction of salary if the SWAT team that went to a hospital to see if the rules of infection control were not respected, they would say that, okay, this person will not get paid this week. So that's actually how they did it. And it was successful in Israel. I don't know if it would work in other places, but apparently it worked for Israel. What's quite astonishing when you start looking at different carbapenemase producers is that you run into some of the same names of the clones. For instance, if you look at the NDM producers, 147 is quite common. You see sequence type 11, which is related to 258, and you see sequence type 14, and it's isolates from different places essentially. If you look at the VIM producers, you see some of the same. You see 147 here also, and you see sequence type 14, and then sometimes you see some that are different. But regardless of whether they come from different parts of the world or if they have different carbapenemases, they tend to have the same sequence type, which is quite interesting in many ways. And obviously it means we need a more refined tool to look at how related they are on a genomic level. And this is where we have, for instance, the core genome MLST, which is really an MLST times quite a lot. Instead of 7, it could be, for instance, around 2,000 genes you are looking at. So it's a gene-by-gene analysis as an alternative to, for instance, a SNP-based analysis. So it's an alternative way of seeing it. It's of course a question, what is actually the core genome? This is a nice paper by Touchon, estimating that the core genome of E. coli is around 2,000, and then you have quite a lot of other things. You have the IS elements, the prophages, and other things of unknown function. So, but looking at this, they have established core genome MLST protocols, which is for E. coli 2,500 genes. It can be sometimes debatable whether this will change, and it will prove that 2,500 is maybe too high. Maybe there are too few isolates that actually share all of those. But anyhow, it's around that number. For Klebsiella pneumoniae, there was this protocol that was developed at the Pasteur Institute, and they opted for around 700 genes of Klebsiella. Could have been higher, so they made a selection of the genes. So it was a slightly different strategy from what was done with E. coli. They did some work to show what is the cutoff that will define something that will be similar with regular MLST. And basically they found that within one sequence type, you should have maximum 100 mismatches. So it's still, when you were looking at the clonal complexes, it was 6 out of 7, and here it's more or less 600 out of 700. But you can also find some interesting information within that variation you see below 100 out of 700. allelic mismatches. And one of the famous subclones that has now emerged, which I already partly mentioned, is sequence type 131H30RX. It's assumed, and this is again just an hypothesis, that it started somehow around the time when the fluoroquinolones were introduced. This was a uropathogenic clone. It started picking up and became stably fluoroquinolone resistant. It started evolving, and then came the INC-F plasmids, which contain the CTXMs, and you got the expansion of H30RX, which is called the Red Queen co-evolution. So basically, it started from uropathogenicity, went through fluoroquinolone resistance, and ended up at fluoroquinolone and ESBL production. And we have looked into our carriers and also the bloodstream infections. Partly you see that B2 phylogroup, which is the most virulent phylogroup in E. coli, is in our population 50% of the bloodstream infections. Out of those, all of them, sequence type 131 are always B2, and most of the cases, so you can see in our bloodstream infections, more than 40% of them are actually sequence type 131. And out of those, we have a substantial amount that are H30RX. You can also see the corresponding situation in population carriers, where it's relatively more common to have A1 and B1, which are less virulent, and less common to have B2, and particularly quite uncommon to have the subclone H30RX. We also have looked into ways of identifying the subclone with core genome MLST. This is generated in Enterobase. Actually, this was done from PacBio data, but you can do this from regular short-read MySeq data, for instance. What's characteristic of the clone is that it has aerobactin, it has adesins, specific capsular genes, it's always MDR. And here you can see the subclone, you can see what's sequence type 131 but not belonging to the subclone, which is actually quite uncommon to be sequence type 131 but not part of the subclone. And then you see for comparison some that are not part of sequence type 131. And we actually have a workspace in Enterobase where you can upload the data and see if it clusters with the H30RX clone. If you look at identification of subclones within other sequence types, this is a collection we have from Oman of different carbapenemase-producing E. coli, and this is what the dendrogram would look like in Enterobase again, if you look at the standard minimal spanning tree. And you see that you have some clusters of several isolates. You even have one sequence type 131, but just one. And you can, just by looking at this, assume that there may be some transmission ongoing, since you see from the same geographical area, you see the same clonal complex. But this is just showing you the power of the tool in separating those into subclones, where you actually can split up CC10, for instance, into two clades, more or less. And even CC38, which consisted of these two, are also two clades. And you can see one example here of clonal complex 101 with just 5 alleles difference, which is likely to be representing some transmission. But that shows the power of these tools if you want to go even on a subclone level, and this is part of what we are doing to try and find out what we're seeing within the carbapenemase producers of different sequence types. How similar are they by core genome MLST? Looking then at epidemic clones of Klebsiella, this is showing you exactly that, and you can see that here you have those that have less than 100 allelic mismatches. They belong to the same core genome MLST type, you could say. and they are found with this gray border around them. And you can see here, if you look at VIM, KPC-producers, you can see that they more or less cluster a bit closer to each other, those that have the same, carry the same genes, but all of them are relatively closely clustering. And in the case of KPC, there are two different parts of it here. And this is 147, which can come both with VIM and DM, as I told you before. So it depends on what type of resolution you want, but more or less it's even with a tool like core genome MLST. It's difficult to see that there's a clear pattern of those with different resistance mechanisms or those that have the same resistance mechanisms are more similar to each other. There's no very clear pattern on that, and that obviously needs to be looked at even in larger strain collections to see what the patterns could be. We have also looked into a little bit more trying to analyze why there is a difference or what the difference could be between epidemic and non-epidemic clones. We have looked at virulence genes and found that the number is quite few in both groups, with the exception of sequence type 14. We have analyzed prophages and did not see any quantitative differences, but we saw that the CRISPR-Cas was higher occurrence in the epidemic clones. This is contrary, actually, with the sequence type 131, which very rarely has CRISPR at all. So I mean, it's not a clear correlation at all that this should be a general mechanism both in Klebsiella and E. coli, but we saw just statistically that there was higher occurrence in the epidemic clones out of CRISPR-Cas. We have some ongoing analysis now of the role of the prophages, more functional assays to look at fitness when you do prophage knockouts. Since we didn't see quantitative differences, we are interested in seeing if there could be more qualitative differences in prophages, and also analysis in the Galleria Melanella virulence model. This is just something that we discussed a little bit earlier today about CRISPR-Cas as the bacterial immune system, and probably most are aware of this, and theories about how this can protect bacteria against foreign DNA, essentially. And also there is the quite recent paper on the anti-CRISPR, which means that phages and mobile elements can evade the CRISPR-Cas immunity. But Professor Raoul informed me today about the paper I was not aware of, which is very recent, about the anti-CRISPR, which is also the bacteria counteracting this system even. But the point of this is we have started looking into this a little bit in E. coli, and we have not really found any good correlations between mobile elements and prophages and CRISPR-Cas occurrence. And even we have this peculiar situation with the B2 phylogroup group, which contains sequence type 131, which does not contain CRISPR systems, as it seems. So most likely there are other systems playing a role here. And I think we are just at the start of beginning to understand, and of course it's also not enough for us just to map these things in silico and see whether they occur, but there are no simple answers so far, at least evident to me, why the epidemic clones are more successful. And we can even see that the epidemic clones grow slower than the non-epidemic ones. So that's also something that may or may not be counterintuitive. What we're doing now is also to look at the competition experiments, where we do fluorescence labeling, and then we analyze the facts to see if this growth rate phenomenon of non-epidemic seemingly being fitter is something that's even observable when they are competing directly against each other, or whether it changes under those circumstances. But it's still somewhat of a conundrum why the epidemic clones are epidemic. And I think there may be a variety of answers. It's not like one answer, it's more or less like cancer or something like this, that there are so many different answers. So strains may become probably epidemic for a number of reasons. With sequence type 131, it can be due to the virulence genes and possibly the fact that it can prevail in the microbiota for a very long time. But for some of the others, it's not at all as clear, and it's certainly not coupled particularly to either virulence or fitness as far as we can say so far. So why is this then useful information, and what can we use this information for that you have been listening to now? I think one of the things that we are quite interested in now is this scenario of having an unexposed individual being colonized with resistant strains. What happens then when there are shifts in the microbiota, and these are really shifts that we need culturomics to detect. We need to do quantitative cultures, we need to measure the relative abundance of the ESBL clone relative to the others, and to see what's actually going on. But what is seemingly going on is that somehow some clones of E. coli, like sequence type 131, dominate the microbiota after some time. They prevail in the microbiota for very long times, and they sort of take over quite a bit of it. And that could be what leads up to infection. We have actually some funding now from JPI-AMR to look into this process and which things could affect it in patients. So we will include patients and follow them for a while in this project according to this hypothesis. And essentially this is so far parts of the hypothesis that's in my mind at the moment, that you can acquire a non-epidemic clone. There will likely not be domination. There will be a short carriage time, and the carriage will be resolved. Whereas sometimes when you acquire instead epidemic clones, you will have long carriage, domination of the strain in the gut microbiota, and this will translate, particularly if it's a uropathogenic strain of E. coli, for instance, you will have multiple infections, either UTIs or even bloodstream infections. Maybe this process is also influenced by antimicrobial consumption. Most likely we can already now stratify carriers into high-risk and low-risk carriers, which is quite helpful, because when it's 5% of the population, it's very difficult to manage that problem, because there are so many carriers, and it would really exhaust all of the resources. So we need some more precise tools, and one of them is, when we have identified an ESBL carrier, to actually try to look into whether this is a carrier of an epidemic clone that you could assume to be able to give these long-term problems. Maybe we can also make therapeutic interventions, and we should definitely then monitor, if we are doing such interventions, we should monitor the clones occurring in bloodstream infection, and this is what we are doing now, or we are going to start using that as a routine tool to do whole genome sequencing of all ESBL occurring in blood, which I think is important, or could be an important way of following the trend. And I also maybe didn't mention that, but this is really our only big AMR problem in Sweden. So apart from this, we have no basic problems. We have tried looking into other ways of approaching this, and other ways to approach it by looking at risk factors for having a bloodstream infection with resistant strains. This is an abstract that we just sent for ECMID, where we looked at retrospectively through the patient records, and we found that previous carriage of cefotaxime-resistant E. coli was the biggest predictor of having bloodstream infection with cefotaxime-resistant E. coli. Also, prostate biopsy and hospitalization abroad were slightly weaker. We did not see any significant association with antimicrobial consumption, with the use of urinary catheter, or with immunosuppression. I think this tells us, at least partly, that in Sweden, previous carriage would mean that the patient has been hospitalized, because otherwise we would not have done a screening for ESBL. So in this case, many of these individuals could have acquired a carriage during hospital stay, and what remains to see is if we can refine the system by looking at how many of them actually also had epidemic clones, which is what we are also doing with the same material, and which will help us more understand this, because these scoring algorithms are quite cumbersome to use in many ways, and also we have tried scoring algorithms proposed in the Netherlands and Italy, and they didn't work at all in our material, so they had very poor predictive value for the risk of having resistant infections. Also, finally, we have seen in travelers, which we have investigated, that they have frequent acquisition of ESBL, but very few of them have phylogenetic B isolates. And these also tend to have short-term carriage. So in conclusion of all this, I have tried to show some of the evidence pointing towards the role of successful or epidemic clones for dissemination of resistance. And of course the carbapenemase are in a particularly important place here, also because many of the carbapenemase-producing strains are becoming Colistin-resistant. We have tools such as core genome MLST and even SNP-based approaches that we can use to refine resolution within MLST clones, and by doing so we can even find a higher level of precision and see other patterns that we could not see when we were just using MLST. We probably need some work to define and make a terminology or something to recognize the important subclones even. And then we still have a lot of work to do to establish some of the reasons why some of the clones are epidemic. I think this will be quite a challenging task, but we are still quite interested in finding, at least in some of the most important clones that I have mentioned, to see if we can come a bit closer to understanding why they are suddenly expanding, and sometimes they are even disappearing without us understanding really what's ongoing. But we have the tools now, so it's quite exciting times to do this type of studies because the technologies have really enabled us to do this more in real time, so we do not always need to carry on with microbiological archaeology, which we have done for decades, because we have not had the tools of really doing this in real time. Rather, we have published papers 5 years later about what was ongoing at that time. And that's really maybe not going to help us enough. And that's my final remark. Thank you very much for listening.
Well, just to comment.
Yep.
One is that we have done work on what I call bad bugs, which are the worst clone in the human history. Usually they lose plenty of genes compared to the epidemic clones get much less genes than the other. And mainly what they lose are associated with metabolism or regulation. So usually when I'm teaching pathogen, I say that in spite of what was the main hypothesis 30 years ago with Falco and Sansonity and so on, that they acquire virulent genes, which is usually not the case at all. They are losing genes, so they are stupid in the regulation. And this is why it's very hard to get a model, because one of the major things that make a bug become pathogen is when it stops to receive the message from the cell that they should not multiply anymore because there is no more food. And this is the whole regulation of the transcription, which is the most commonly lost things in the. For example, very interestingly, what make E. coli very, very aggressive is losing, of course, lactico dehydrogenase gene. So it's losing gene that make a Shigella, for example. Shigella is stuff like this. So I think the most important thing that people, it's so hard to come back from these studies that the bad bug has acquired virulence genes, you need to be very careful in looking more what they've lost compared to what they've gained.
Yeah.
And including gene with a known function, which is very difficult. The other thing is that it is bizarre. This is why we are discussing since years with Jean-Marc on the role of CRISPR. But it's true that the clonal expansion is usually associated with the capability to acquire resistance. And then the phenotype that we had for long to identify the clones, well, of course, was wrong. This is very clear for type 1, for example. And usually it's associated with the fact that they lose one or several CRISPR. And so the clones are commonly associated with the loss of CRISPR that make it possible that acquired sequence from the outside and become or not resistant. So I think it's a general mechanism. But I think that the very beginning is starting by losing genes to become virulent.
Yeah, no, I think those are really really good comments. And with regards to the first one, I think one of the puzzling thing is that I think what you're saying may be actually true for instance in the Klebsiellas we have looked at, because they seem to have, although we have not done competition, we have done other fitness experiments to indicate that they actually have lost fitness, the epidemic ones. But I think it's the other way around with this E. coli subclone. So that's strange in many ways.
In vitro.
Yeah.
Just remember, because I'm French, I need to tell something about Pasteur. You know, the vaccination was based on the fact that they used the coll agent, which is Pasteur and grows it in agar, and after a few passages in agar, he started to put that back in the poultry, and it was not efficient anymore. So it was the basic of the vaccine.
Yeah.
So that means what is fits in vitro and what is fit in the chicken is different.
Yeah.
And what is fit in humans, it's very hard because it's not a chicken, it's not a mouse, it's not an animal model, and it's not agar. So the only knowledge that we have on fitness in human is that it makes you sick or it's multiplied.
Yeah, yeah.
So we don't have any model out of it. So that's true that when you look in agar, for example, it's not traducing. If you get several plague is like that.
Yeah.
If you pass that 20 times, it loses plasma. It's more efficient. So it's very hard. The fitness definition and the virulent definition is very, very difficult to become.
It is, and particularly those non-acknowledged genes that could have importance for virulence. I mean, you can search in silico for many things, but it will not tell you the whole truth about it. But I think the in vivo virulence models can probably be useful, but they are still models, and they are not human.
Toxin is fact.
Yeah.
I think I believe in toxin. I don't believe in anything else. But toxin, yes, I know what it is. You give that to mouse, it's dead. You put the antitoxin, so it works in humans. Toxin is fine.
Yeah.
The very first thing that at the beginning of with Michel, the beginning of the genomic study, at the beginning of the 21st century, when we check, the more virulence bags get less virulent genes than the non-virulent bags.
Yeah.
Well, that's true.
Yeah, it's very interesting, but I think in terms of E. coli and Klebsiella, it may also they are very different in so many ways, and with ecological niche and everything. So maybe it's not entirely without logic that they are behaving differently in this sense, also in terms of what is successful. At least in our particular situation, Klebsiella still usually infects somehow immunosuppressed individuals or neonates. So it can be a different story. They can cause infections either way, whereas E. coli, they have also the challenge of infecting healthy individuals. And so there may be different explanations for why some are successful in one species, and there could be other explanations in another species. Another bug we are planning to look into is also Pseudomonas. But there's of course some parts known already there about the type 3 secretion systems and so on. But but there can still be more things to look into.
Did you have, using your genome analysis of the new ST14 in Klebsiella,
OST14,
genes came in from Asia, responsible for liver abscesses?
yeah.
You have in your analysis done this, or you do not have any strains from
We
those
don't.
particular?
No, we don't have any strains that we know have caused PLA, but they have some of the same. They have serotype K2 and they have the sequence type that's known to give that. So I think the sequence type 14 Klebsiella is slightly different from the others we have seen here. So that one seems to be more virulent. But the other ones, like 147 and 258, seem to be different in that context. But we don't exactly have any strains from PLA patients in the collection. But I would assume that those sequence type 14 isolates are among those that could give that. Although the PLA may also be something to do with, I suppose, even genetic susceptibility in the host. I don't know if those strains are equally likely to give phylogenetic liver abscess here, for instance. But that's, I think, insufficiently looked into. So I don't know if you have seen here a lot of cases with phylogenetic liver abscess down to Klebsiella.
No.
Yeah. So I think there must be also some, since we probably have these strains here also, there must be also genetical factors with the patients.
Yeah, maybe also
Yeah.
microbial, whatever.
Yeah, it could be something else we don't understand.
Well, thank you very much.